Our cells have four main macromolecules that aid in keeping the cell alive. This helps them function properly. Hey guys, welcome to SciMath, and today we are going to be talking about the structure and function of these large biomolecules that help in keeping our cells alive and function and help them in functioning properly. So let's get started. First, let's talk about macromolecules and what they are. So, these macromolecules are just composed of small organic molecules, and in the term itself, macro just means large, and these molecules make up all living things. Next, there's four main classes of these macromolecules, and they are carbohydrates, lipids, proteins, and nucleic acids. And we're going to talk about each one more in depth in this video. Before we begin that, we need to understand what the main difference between monomers and polymers are. So polymer is a long molecule composed of many similar building blocks linked by covalent bonds, and essentially that means that a whole structure that is made up of small single units. While monomers are the small building blocks that compose those polymers, and mono means one, and poly means many. So that's the difference between them. Like you can think of a single Lego building block, right? That would be considered a monomer, and then when you make your building or structure or whatever it is with those Lego blocks, that would be considered a polymer because it's made up of so many of those similar building blocks, right? So that's the main difference between monomers and polymers. Next, let's talk about carbohydrates. These include sugars and their polymers. And so monosaccharides are the simplest carbohydrates. Monosaccharides are the monomers to carbohydrates. And when two monosaccharides bond together, it's called a disaccharide, and the prefix di just means two. Lastly, polysaccharides are polymers of many monosaccharides. So when more than two monosaccharides bond together, it's called a polysaccharide. Again, poly meaning many. Next, I want to talk about lipids. Lipids are often referred to as fats or waxes, and they have little or no affinity for water because they are nonpolar, meaning that, for example, oil, when you mix it in water, it doesn't dissolve, and that's because water is polar and oil is nonpolar. A good thing to remember is that since water is polar and most things on Earth are polar, like dissolves like, and that's why water is considered to be a universal solvent, but that's not all accurate, that term, calling water universal solvent, because there are exceptions to it, like oil, like lipids, things that are non-polar that don't dissolve in it. But yeah, and lipids are useful both as a long-term energy source and structurally. And so another example of lipid would be butter. Next, let's talk about proteins. Proteins have a variety of functions, including structural support, storage, transport, defense, and movement. Protein enzymes, they function as catalysts in cells, essentially speeding up the reaction in the cells. And so amino acids, in the case of proteins, are the monomers, and they make up polypeptides. And those polypeptides make up proteins. And that's kind of how a protein looks like, all folded up in that diagram. Next, let's talk about amino acids. So, like I mentioned before, they are the monomers of proteins, and they contain both carboxyl and amino groups. So in this diagram that you can see here, there's kind of a central carbon atom, and everything is attached to it, all four sides. So to your left, that's the carboxyl group because it's double bonded to an oxygen atom and bonded to an OH. That's a functional group, carboxyl, and there are more functional groups out there, but it's a whole different topic. Then there's a hydrogen atom that's bonded to it at the top, and to the left there's an amino group, NH2, and then on the bottom there's a variable R group. So there are 20 amino acids that exist, and so these R groups are what make each amino acid unique. And so if there's 20 amino acids, it makes sense that there are 20 R groups. Next, let's talk about nucleic acids and genes specifically. So the amino acid sequence of a polypeptide is decoded by a gene. A gene essentially directs and runs how the amino acid sequence should look like of a polypeptide. And a gene is composed of DNA. But nucleic acids include DNA and RNA. Nucleic acids are made up of monomers called nucleotides, so that's their monomers, just like proteins had amino acids and carbohydrates had monosaccharides. Here I'm going to talk about RNA. Now, the reason I'm not talking about DNA is because I have a whole separate video on that and its applications. So if you want to check that out, I would highly recommend that. But I'm just going to talk about RNA here. And so complementary pairing is also important in RNA, just like it is in DNA. And transfer RNA delivers its amino acids based on base pairing. So you can think of transfer or tRNA, its job is to deliver, right, its amino acids to the base pairing. So in in DNA, there's adenine, thymine, cytosine, and guanine, and A pairs with T, C pairs with G. But here in RNA, the difference is that instead of thymine, there's uracil. So then it pairs up with the adenine. And another difference between RNA and DNA is that RNA is typically single-stranded, whereas DNA is a double helix shape, like a ladder. And so there's two sides, but this is typically single-stranded. So guys, that was the end of the video. Hope you guys learned something new and enjoyed it. If you did, don't forget to like, share, and subscribe. Thanks for watching.